To watch the latest from India Science, don't forget to subscribe to the channel and press on the bell icon to get notifications on all the science-related videos. The Human Genome Project, or HGP, was one of the great feats of exploration in scientific history. It was an inward voyage of discovery led by an international team of researchers looking to sequence and map all of the genes, together known as the genome, of members of our species, Homo sapiens. Beginning on October 1, 1990 and completed in April 2003, the HGP gave us the ability for the first time to read nature's complete genetic blueprint for building a human being. As we celebrate the 20th anniversary of publication of the draft human genome sequence, another genome revolution is slowly evolving in space to explore extraterrestrial life on other planets, to identify the health status of astronauts, and ultimately establishing a molecular diagnostic lab in the International Space Station or in space colonies at Mars. Sounds like a crazy idea? Not anymore. If we have to believe in the research, this is going to happen in the near future. Many countries, including India, are vying for space in space. The race for space colonization has already started. NASA's Mars 2020 rover successfully touched down in the Jezero crater in February 2021. NASA is planning to send astronauts to orbit Mars and return them safely by the mid-2030s, and a manned landing on the red planet will soon follow. Private players like Elon Musk and Richard Branson are making spaceflight exciting by their space tourism and space colonies projects. These missions represent a new era in our search for life beyond Earth. And searching for life means looking for the presence of nucleic acid, either DNA or RNA, through sequencing. Sequencing is the process of determining the nucleic acid sequence, the order of nucleotides in DNA or RNA. It is surely a challenging task. Major challenge in sequencing nucleic acids is in pipetting, that is, transferring measured reagents and managing bulky instruments that we use in molecular biology labs. But these challenges were overcome in 2016 when NASA scientists made a breakthrough by sequencing DNA in space inside the International Space Station. NASA astronaut Kate Rubins successfully sequenced DNA in microgravity as part of the biomolecule sequencer experiment. They used a tiny device called the Minion, developed by Oxford Nanopore Technologies. Nanopore sequencing is a unique, scalable technology that enables direct, real-time analysis of long DNA or RNA fragments. It works by monitoring changes to an electrical current as nucleic acids are passed through a protein nanopore. The resulting signal is decoded to provide the specific DNA or RNA sequence. In 2017, NASA astronaut Peggy Whitson collected and tested samples of microbial growth found around the ISS. Alongside Minion, astronauts also tested Mini-PCR, a thermal cycler which is used for DNA amplification. Using these platforms, she identified unknown microbes present around ISS. In 2018, another astronaut, Ricky Arnold, sequenced RNA. With the sequencing of DNA and RNA in the International Space Station, scientists demonstrated that sequencing is possible under microgravity conditions, setting forth a new era in space genomics. So what's next? A space-based DNA sequencer would be an important tool to monitor the health status of astronauts, especially during long-duration missions like journeys to Mars. It could also be used to identify DNA or RNA-based life forms present in other planets. Scientists like J. Craig Venter are already eyeing on the potential of genomics in space. His team at Synthetic Genomics, led by Daniel G. Gibson, has already developed a digital to biological converter or DBC that accepts digital representations of DNA over the internet and reconstructs them on the spot using the chemical building blocks of life, that is adenine, cytosine, guanine, and thymine. Like a printer, it needs cartridges, but instead of colors, it needs bottles of chemicals. It's packaging complex biology that each of our tiny cells do remarkably well at a much smaller scale. Venter hopes that DBC technology could be used to develop biological teleporters for sequencing the Martian life or even recreate it sitting on Earth like a fax machine. This technology is cheaper than bringing biological samples from another planet and studying in our labs. This will also reduce the chance of contaminating a planet with unknown organisms. This new technology is also faster. Since it uses digital information, it can travel at the speed of light and reach Mars within minutes, while it takes months to physically reach there. When space colonies become reality, we can use this technology to provide personalized healthcare for our brothers and sisters settled in other planets by referring our Earth doctors. Indeed, genomics is making great strides for the next-gen humans in space colonies.